data, when we have the genomic DNA and we want to infer the information which is encoded there in the form of ATGC. So we want to identify those sequences, nucleotide sequences, that their arrangement, ATGC, how they are arranged there. So suppose our interest in any genomic DNA from any organism, right? So that genomic DNA is isolated, and then from there we prepare the DNA library. That DNA library is prepared by using the random shearing, like physical shearing or the enzymatic shearing, and then we prepare the random library of that particular genomic DNA which we receive. That may be in the length of 200 to 600 base pairs, 500 base pairs in length. That is used to ligate the adapters for on this sequence, on this template DNA, which we prepare the library and we site select it there. After that, the ligation of these adapters, we do the flow cell, we apply it to the flow cell, and on that flow cell, bridge amplification is performed. On that, in that bridge amplification, each template DNA is multiplied, that is into multiple copies, and then after that they are applied to the sequencing platform there, where the sequences are obtained. So how the sequences are obtained here, like each nucleotide is labeled with the fluorophore, and when they are attached, they are synthesized there, they are used in the synthesis there, a laser light hits there, and then that laser light produces by the fluorophore, that creates the emission of the light from there, which is recorded by the detector, and that detector records that particular wavelength, and it says that it is A, this is T, it is G, right? Here at this level, when the base call is performed, like how the bases are arranged, which base is incorporating at that time, that is called the base call. And that time, if the intensity, signal intensity is good, that is also recorded as in the terms of the quality. So what we receive here from the next generation sequencing platform, such as Illumina workflow we are seeing here, what we see here, what we receive, that is the sequences in the form of ATGC, how they are arranged, and along with that, for each of the bases, we receive the quality of that. Looks like something looks like this, right? So in each file there are millions or billions of sequences we receive for each genomic fragment we sequence, right? So each sequence is represented by, in this FASTQ file, by 4 lines. First line here, that is the header for the sequence. So that starts with the @ sign and then the identifier, sequence identifier, and then if it is a paired end or singlet, they are also 1 by 2 or 1 by 1. The same way, the next line will be the nucleotide sequences here, and then the third line will be starting with plus sign, means it is the quality value sign here, quality value header. Then the next fourth line is representing the quality of each base which was recorded there during the base call. So suppose this is A and this is A. So this is not the base here, it is representing the quality of that particular base here, A base. Right. Is it clear to you, this FASTQ and then the workflow?
Yes, sir.
Great. So, right. How the quality values are interpreted? These are in the terms of the characters, what we see here, like A, B, B, F, F, whatever is written there, or in the fourth line. So how these values or these characters are converted to there. So each quality value has the Phred quality score, right? So this is a unit of quality identification of the nucleobases generated by automated DNA sequencing platform.
Hello.
Yes.
Yes, sir. What 1 by 2 denotes?
So that is the, if it is a paired end data, right? So it will be 1 by 2 or 1 by 1. So forward for forward file, it will be 1 by 1, and for the reverse, it will be 1 by 2. I will show you that when we see the files. Yeah, it will be much clear to you. It depends. Like there will be the library name, like sequence identifier, and then there will be how the sequencing, which platform was used for the sequencing. So, but this data was obtained from the SRA database, right? So I
Single
will
end
explain to
data
you.
represents for what?
Which one?
Single end read.
Okay, single end reads, they are coming. I will explain to you in the next slide. Okay, I think it will be clear. Then we can discuss there. Right. So when we have the Fred quality score here, that is the unit of quality identification of the nucleobases generated by the automated DNA sequencing platforms. Right. Fred quality scores are assigned to each nucleotide base call during the sequencing process, during the base call step, when I showed you at this step. Right. So they are assigned there. And I told you that if the signal is good, they will be assigned good quality. If the signal is not good, they will be assigned lower quality. So how the Fred quality scores are interpreted here? If the Fred quality score is 10, then there is the probability of incorrect base call of 1 in 10. Like there are 90% accuracy and 10% inaccuracy. If the Fred quality score of that particular base position is 20, it means it is 99% accurate. 30, 99.9%, and for 40 it is 99.99%. So what does it mean here when we talk about the Illumina dataset, right? So in the Illumina dataset, for the Illumina platform, they claim that it has 0.01% of error, right? And that is the limit of Illumina platform. So we get the maximum sequence quality of the from the Illumina platform, that is 40. We cannot go 50 and 60 output from the Illumina platforms because they have the limit of 99.99% of accuracy. Right? Is it clear that accuracy is there? So what we see, the base calls like the Fred quality score there, they only assign up to the 40%, like accuracy. The Fred quality score 40. Right. So how the first Q quality values are interpreted? Like there are different types of the encodings available, right? Like Sanger here, see Sanger for the Fred plus 33. Raw reads typically 0 to 40, so 99.9% accuracy. So Alexa minus 5 to 40, right? So 99.9% accuracy here. Like the other reads also here, you can see that their accuracy is, or their different encodings. So what does these encodings mean here? Suppose we are using Illumina 1.3 plus sequences or the output. So in that case, the quality value will start from here at 64, right? For this Illumina 1.3. So it is not the 64, it is the ASCII character which has the value of 64. You might be knowing that each character like A, B, C, D, they have the ASCII values. Like here the caps and the in the large A or the caps A, which has the 65 of the ASCII value, right? The same way B has 66. Like those ASCII values are written there. So that is A here denoting the value of 65 here, which starts from 0 here and goes up to 40. Then the same way, Illumina 1.8 plus Fred score, which starts from 33 here, 0, and it goes up to 41, right? So if the quality is 0, we will see exclamation sign. If the quality is 41, we will see I written there, right? H will be 40, G will be 39, like that in the quality line there at the fourth line, in this line. H, H, E. So it means that if it is 1.8 plus data we are seeing, it means it has a good quality, like G, H, I. A is also good, 30 plus. So 99.9% accuracy is there, more than 99.9% accuracy. Is it clear to you? This Fred score encode, Fred quality score encoding. So there are different encodings depending on. So it doesn't matter what encoding is being used. It goes from 0 to 40, 50, or up to 60. Right? Is it clear?
Yeah, it's clear.
So this is a chart which is showing the ASCII code of each character here. like 33 for exclamation, dollar sign for 36. And the same way, if we see the A B C D letters here, so we see here B for the uppercase B, which is 66, E for the 69, like that. So these are the ASCII values. They are encoded into different methods of the quality value representation. So when we talk about the paired end and the single end sequencing, right? So here we see the paired end sequences, like when we sequence the same template DNA from both the ends, right? So we have the primers and we sequence it from the 5 prime, and then from the reverse side also we sequence it. Then that is called the paired end sequences, and it covers the whole template sequence here. But in the case of single end DNA sequencing, it will be only forward read sequence. We will not have the reverse read sequence here. What is the benefit of using the paired end data here? That it gives us the more robust assembly output for the repeat regions. So repeat regions are assembled very well when we have the paired end data. Along with that, if we have the sequencing gaps available during the assembly process, so those paired end reads can be used to identify the joining sequences, and then we can fill those gaps with ends if the sequences are not available. If the sequences are available, then we can use those particular nucleotides to fill those gaps, right? So this paired end data is better than the single end data, but still we can use the single end data if we have only single end, right? So first we will see here, when we receive the data from the sequencing platform, we have the raw data. We receive the raw data if the sequencing provider hasn't processed it for the good quality. So we receive it as the raw. So there might be the errors available present in the data. What we need to do next, we have to check the quality of that data, if it is of good quality or not. If it is not of good quality, then we can remove the bad quality bases and then the sequences as well. So to check the quality of these sequences, we can use here FastQ, FastQC toolkit here, right? That can be used to check the quality of the sequencing reads. Once we know that if it is not of good quality, then we can proceed for the trimming of those particular bad quality sequences and reads from there, right? So here we will be using the Trimomatic tool to remove the bad sequences and the base pairs which are not of good quality. Tribomatic uses here the sliding window approach. So it will take, I will explain it to you more in the detail when we will be working there. So it uses the, like for the sliding window, it takes the 4 nucleotides at once and then look for the particular quality. And if the quality doesn't match there, it will trim the sequences and then take the next 4 bases by sliding 1 nucleotide. And then by sliding the next 1 nucleotide to take the next 4 nucleotides. Right. So that is the more enhanced and then the better approach to check the quality of the reads, right? So do you have any questions? You can ask and then we can move to the terminal. I will share the window.
It's fine.
I will go to the directory where the data is available. Here we see FASTQ sequences for the forward and the reverse for the Mycoplasma pneumoniae genome, which I downloaded from the NCBI. So I will show you how the FASTQ files look here. So you see, this is the header, FASTQ header sequences, then the quality header, and then the quality values, different quality scores here, which are written for each corresponding base here. Right. This is the sequence read number, and then this is forward. Forward, one means forward, right? You will see like the sequence identifier, and then there is like colon, and then one. Like you will see the different types of the formats written there when you receive the data from the sequencing platform. But the main mainly you will see here one for the forward, and for the reverse you will see two, right? So we can run here fast QC to check the quality of this raw data here, what we have. Okay, I will show you how you can install it. So there are different ways to install fast QC toolkit. So one installation is available from the Anaconda. So you can do one thing like conda install -c bioconda and then fast QC. Right. So if you run this command, it will install the fast QC in the Anaconda installation, and you can run it whenever you want to use. The other way of the installation is sudo apt install fast QC. For this, you need to have the password of the system. You need to have the sudo password, which is your login password if you are the administrator. If you are not administrator, you need to ask your administrator to install it for you. Right. Once it is installed, you can check the help of this fastqc tool using fastqc --help. Right. So we can see there are various different options available here to run the fastqc tool. There is we can check the version of the tool by running -v. We need to define or provide the output directory. So we need to first create the output directory, and then we give it to the fastqc toolkit that save or create all the result files inside this output directory here. Right. Cassava tool is there, so we can use the Cassava tool output to the fastqc directly. Nano is for the nanopore sequencing reads. If we have the nanopore sequencing reads in the FAST5 format, we can use them directly here by providing this option that it will identify those reads as FAST5 format and use it for the quality analysis. If running with Cassava, then don't remove read flagged by Cassava as poor quality. So check the quality and don't tell that they are of bad quality already. So check the all of the reads there. Right, extract zipped output of that result files which will be created. Right, Java path, if you are running it with your own installation and you want to give the Java path there, you can use that also. You have to provide the Java path if it is not in the path there. Right, no extract. So this is like output files should be compressed. Do not uncompress the output after creating it. So there are various different options available, like minimum length of the reads we can assign here. Let's take the 50, 60, whatever the length we will define. If the reads are smaller than that, it will omit those reads for the quality analysis. Format of the data is BAM, SAM, MAB, SAM, MAB, or FASTQ files. So we have seen already the FASTQ format there. but there are different formats like BAM format we can also use here for as the input. Number of threads we can assign here. If we have multiple threads available, we can use them. We can also define here or provide the contaminants file. So if we suspect that there must be contaminants available in your genomic data set, then you can use those sequences like that particular known genome. You can provide here as the contaminant file, and it will look at those sequences by aligning to the contaminant data that and remove or tell that how many sequences are contaminant there in the data set. List of adapters you can also provide here. If you know that these adapters were used during the sequencing, you can use these adapters to identify in there. K-mer based approach is also available, so you can define -k option and then provide the between 2 to 10 nucleotides of the K-mer size you can define here. Default is 7, right? So let's create here fast QC. Okay, mkdir. I will create first the directory fast QC out. Right. All the results will be saved in this fast QC out directory. Right. Fast QC -o and then fast QC out. -p we can use here 3, 4, and then the read names. So we have here 2 reads, forward and reverse. We can supply them together one after one. So we can use any number of reads here to check the quality. So I'm using here forward read and then the reverse read. Right. It will run and store all the results in this particular directory.
Hello.
Yes.
Hello.
Yes. Yes. Okay. Wait. I will paste the command. So you need to create your directory here, right? This fastqc out you can create, and you need to have this file also. Okay. So I will share this file today, so you can use it. Copy this and send it to you.
Can we download this FastQ?
Yes, you can definitely. Yeah. So you can download it from the SRA database, from the NCBI or any of the nucleotide database deposition databases. So the easiest way to download any this fast QC file from the SRA database, I will show you. You can let me share the screen first. The NCBI website, you have to go there and log in, or you have to use their fast QC. I think it is FastQ dump toolkit, right? So you have to download that FastQ dump, which is available from the NCBI. You can type here as accessor number. So the accessor number I will type here, the same which we are using the data. You can write here the same data. And you can download these short reads from the ENA short SRA database here. So which is connected to the NCBI as well. So you can download these here by selecting these files and then download selected, or you just download all. You can click on any of these. It will start the download for you and you will get this data. This is compressed format in .gz. So you have to run the command to uncompress and then you can use it, right? So this is the same data set which I am using right now here. So output is created here. It was finished. I think it is shared to you. Yes. I'm inside the fastqc out result directory. We have this output here. HTML1 for the forward, for the reverse, and then the forward HTML. We can open it any of the web browsers like Google Chrome, Mozilla Firefox, or Microsoft Edge. Any of the browser can be used to open these HTML files. So I will go there and then share the screen from the Windows side. Here, let me share the screen first. We are seeing the quality or fastqc report here, which was generated by using the fastqc tool. We see here on the left side pan summary. So basic statistics, per base sequence quality, per sequence quality score. And the basic stats what we see in here, that is what is this data. So the file name of that data, it is forward 100,000 sequences. Then the file type is conventional base call. Illumina 1.9 encoding here. So what encoding is used for the quality, for quality values, that is identified as 1.9 Illumina. Total sequences are 100,000 and total sequences flagged as poor quality. 0 sequences were flagged as poor quality. Sequence length is in variable reason, 35 to 250 base pairs. Percent GC is 41. What we are seeing here, these crosses means there is the serious issue, or this has the problem with the data, like per base sequence content has a problem, which shows here cross. Per base sequence GC content, it has the warning sign or warning output here in the result, like it doesn't look good but it's okay, still it works fine. Sequence length distribution, it also has the problem, may have the it may have the problem, but it is not the case here. Over represented sequences, it has the issue. The green ticks here saying that the data is fine, like at this output or in this analysis. Like basic stats, it's okay. Per base sequence quality, it looks okay here. So what this graph showing here, on the x-axis there are the base positions starting from 1 to 250. Like it is in the range of 35 to 250 base pairs. Then y axis here showing the Fred quality score. So that is the mean Fred quality score at each base position. So at base position number 1, mean base quality score here, it is 34 or like 33. So what is 33 here? It is the blue line saying that this is the mean quality score here, and this is the median quality score of the data, red, line here, and this is showing the error bars, like how much is the deviation there of the data in the quality value, right? So if the quality value falls below 20 Fred score here, 20, it is a issue. It's an issue here. We cannot accept that, and we need to trim those sequences or remove those sequences which are not completing the quality score of 20. So this looks fine here. This looks okay, right? At the ends even it is going down, but it still is not going below 20 Fred quality score, so we can accept it. But most of the times people ask about the sequencing, so Q20, Q25, Q30 output. So whatever the sequencing provider is giving, the data must not go below the 30 quality score from this, or Q25 means it should not go below Q25, right? So this is okay. Per base sequence quality. Per sequence quality scores so the same output here, like the data, but the interpretation is different. Like how many number of sequences are qualifying a Fred quality score here. So mostly they are better. Like very few sequences which are having the quality score here below 28 or 27. Very few sequences we see in this region. Otherwise, they are zero. And if we add more sequences, then it goes up here up to 38. Right. So it says that the data is of good quality, and we see here the green tick also. Per base sequence content, it has the problem because all of the base positions here, like starting from 1 to 250, it should be following the same trend. Like there should not be any over represented or down represented sequences bases, right? Like percent T T A G, there we see here it is higher at this end, and at the 3 prime end also it is showing over represented and down represented bases here. So what can be done here, we during the filter process or during the trimming process using the tribomatic, we can chop these reads off here at this side at the 5 prime, and then at the 3 prime also we can chop off here at 240 or 245 base pairs. So that will remove these bases from there, and it will be okay. Per sequence GC content, it also has the issue. Like this is the theoretical distribution represented by the blue peak here or blue graph. And we see here the red one here, which is the GC count on the basis of the reads, like for each of the reads, and then the final mean GC content here. And we see here, which is showing a hump here, but it's still in the same range and not much bigger, like bigger hump we see or any different two peaks we don't see here. So it means that this is okay, like it is in the range of 41% or something, 45% of the GC, which matches the organism's GC content as well. Sequence length distribution per base and content, sorry, this is the how many ends are present. So and why the ends are incorporated during the sequencing, I told you, when if the base call is not perfect and sequencing platform or that it was not detected there, like it has a what base it is, so it will assign that particular base position as the end there. So in this data, there are no ends present, but it is not always the case. Most of the times we see various ends present in the reads. So we can select like we don't want any end, or we trim the ends where it encounters. So like 1, 2, 3, how many ends we can allow in the data set. So that is also okay. So if this data set doesn't have, if your data set has it, you can remove those ends using that filter option. Then there is the sequence length distribution. So it says that like majority of the sequences are in the range of 240 to 251, and very few sequences are in the range of like smaller than that. So it says that that may be an issue, but it's not the issue here, because we know that the reads are of variable length already. If there was some problem with the data, like you process it or you are having any issue with the read lengths, then you can talk to the sequencing provider or the technician who has performed the sequencing. Sequence duplication levels. So why it is important, like when we are doing the PCR amplification, like those sequencing protocols, then in that case we might see the higher duplication levels. But if the library was prepared, random library was prepared, and then the normal workflow was followed, then in that case we must not see the higher number of the duplicated sequences, as the case we see here. Like it is number of the percent of the duplication, like 1 2 sequences, and then this is the duplication label, like how many sequences are duplicated there, right? So there are not many sequences which are duplicated. Over represented sequences, so we see here one sequence which is over represented in 175 reads, and that might be potentially a true seq adapter, index 1, 100% over 50 base pairs. So that may be a contaminant there which remains after the processing or after the data which was collected from the sequencing platform. So it must be removed before going further analysis. Adapter contents looks okay. It doesn't have any adapter available there. How many adapters are available in the FastQC toolkit? It did not match to any of those adapter sequences. So it doesn't have any adapter here. There are a variety of adapters like Illumina Universal, small RNA 3', small RNA 5', next Tera transpose sequence, and solid small RNA adapter is also present there. So none of them are available or present in this. So what we have seen in this data, and we can select what parameters we need to select during the trimming option step. So first thing is to trim the reads here, at least at the 50 nucleotides on the 5', and at least 245 base pairs from the, so total length will be 245 here, right, for the 3' trimming. Then quality looks okay here. We need not to select anything for the forward read. For the reverse read, if we see, the quality still looks okay, right? So if it goes below that, we can select trim the reads which are having bad quality. So as we see here, the deviation or the error bars showing much more down in the quality at the last base position here. So we can select this particular base position, or at the end, that this should be removed. So we will select that particular quality, like of 20, or maybe we can select the 25, right? Then everything looks same for the as the forward reads, except this one here, like this is also same. So we will take care of this by trimming these positions here and from the 5' too. Per sequence GC content is the same here, right? Sequence length distribution, ends are not present here. Per base and content is also, no ends were recorded. Distribution of sequence here, the sequence length distribution, that is also the same as for the forward reads we have seen. Sequence duplication level is also same as we have the forward reads. So not many sequences are duplicated. Over represented sequences are not present here in the reverse read file, and no adapters are also identified, right? So we have two parameters to select to improve the quality of the reverse read also, and then the trimming the reads here at the 5' and then the 3', right? So I will share the screen for that. Do you have any questions for this output and the results?
Sir,
Yes.
hello. Sir,
Yes.
from where did you get this report, Fast QC report?
Pardon?
Where did you get this, like,
Oh
this
yeah,
report?
so I think you missed it. I
Yeah.
will show you. Yeah. This is here in the fastqc out. Right. I ran this command fastqc after installing it. Fastqc, and then in the output directory -o for the output, create the results in this fastqc out, and then number of threads were used 3. So it doesn't matter if you have more, you can use not 3, but you can use like 5, 6, 8, 16, how many threads your PC has. Then the next is input files we are providing here. So this is the forward file and then the reverse file. Okay. After running this, we saw the output in this fastqc directory, this HTML file. Okay. And I opened it from the Windows side in the Google Chrome to visualize it. Okay. So this is the output from the fastqc tool. You can also use the fastqc tool in the Windows. So exe file is also available for the fastqc. As we have those parameters to select to process the data, we can run Tremomatic. To install the Tremomatic, we can run a command. Yes.
When we do trimming, then is there any chances of losing some information?
Yes, definitely, because it will remove those
How
bad
to
quality
work on
sequences. Okay, but
and then
the
how
fact is
we
that those bad
keep
quality sequences are not of any advantage for us. They may create the
so
trouble because they are not having the good quality. They are not the same as we wanted them to be. So it's better to remove the bad ones rather than getting the wrong information by using those wrong ones. Okay. So we will lose, but they are not of any use.
So,
Yes,
the reverse file and forward file are two different files that we have to download from the NCBI or EMBL.
sure. Yes, yes, yes.
Okay, sir.
So what I have done here, I have subsampled it to reduce the number of reads in each file to 100,000 sequences in each file. Okay, so you can use like head command when you download the file. You can use head minus 400,000, 40,000, 400,000, and then the file name, which file you have downloaded, like this one, forward dot fq or fastq, whatever is there, right? And then create a file like this, 100,000 dot fastq, right? Like that, you can subsample it from both the files for forward and the reverse. It will be forward. We can create two files having the same organization of the reads in the forward and reverse, because each reads are having the same organization, like at what position line number there is, that sequence is present in the file 1 or forward file, at the same position or line number will be present in the forward reverse file. So they are corresponding sequences kept in both the files. If you mess up with the organization or the position of those reads in both the files, that will create a problem for you to, for any of the tools to identify where are those reads, right? So they must be in the same line number in both the files. To install the Tremomatic tool, you can run the command here, conda install -c bioconda, and then Tremomatic. You run this, it will install it in the Anaconda distribution. If you are using the Anaconda, if you are not using the Anaconda, then you can go to this particular page here. Let me share that particular Tremomatic page. So you can come to this place or this web page for the official Trimomatic tool. You will find here the manual for the Trimomatic, and then the binary source file is available also. So if you click on the binary here, you can download the jar file for the Trimomatic zip, and then you have to unzip it, and then you can run the Trimomatic from there. Otherwise, you can install it using the Anaconda or Conda command. Conda install -c bioconda and then Trimomatic. Right, you can check the manual here, which is also listed. You can go to the GitHub page here as well for the version 0.40. If you are interested in the latest version, you can download it from the GitHub page here. The link is given on this page. Right, once you have installed the Trimomatic, you can run it. Right. So here are two files we have to run the Trimomatic on it. I'll copy the command which is given in the file here. I will share this file with you. Right. Here is the Trimomatic help. You can check it. Not much available here on this help page. So you can check the Trimomatic manual for detailed informations for all the detailed options and the flags and their uses. Here you can use the paired end or the single end input files. So if you have the single end data, you can use SE. If you have paired end, you can use PE. Then we can use the number of threads to maximize the process, like parallelize the process in the same run. Thread quality is score here, thread 33 or thread 64. So it is thread 33 coming from the Illumina platform. So it uses thread 33 because it is Illumina 1.9, and we know it from the fast QC report there. As I showed you that it shows 1.9. So that encoding is thread 33. Then the trim log file we can create here if we want to save the output stats. Summary of the stat there. We can also create a summary file output. Quiet, don't show anything on the screen, run it behind there. Validate the pairs forward and reverse before trimming the reads and then removing the bases. And then there will be the base in means input file 1. Sorry, this base in is for the, if you have multiple files, you can provide the input base names for the files and it will be recognized there. If you have the, then you have to put the input file 1, input file 2, forward and reverse files. Base output file will be here. So output base name, then output file 1 paired, output file 1 unpaired, and output file 2 paired and output file 2 unpaired, and then the trimming options. Right. So let me show you this one. Here it first. Here I'm using paired and data. So Trimomatic PE. Number of threads I'm using here 4, and then forward read fast queue 100,000 forward and reverse fast queue for 100,000. And the reads are in the same organization in both the files. Then there will be the output directory if you want to create. So you just create a directory and then run, give the output name here. So it will be the forward paired. We can change the name here to forward paired or forward P, right? then there will be forward unpaired. So the reads which are not of good quality in the reverse file, but they are good in forward, they will be retained in this file because their pairs were removed from the reverse file. So they will be kept into a forward unpaired file. For this paired output, their pairs will be kept in the reverse file. Right. The same way there is reverse pair. So this is the same paired end files, forward and reverse pairs. And these are the unpaired files for the reverse one also. That forward read was not good, so that was removed. And reverse file or reverse read was good, so it was kept in the unpaired file. Right. So that is the output. Now we use the Illumina clip here. So I have downloaded all the adapters which are available for this tool here. So you can download it from the, I think it is available from the Trimomatic GitHub page, right? And then you can give the alignment option here. So two mismatches are allowed, 30 alignment for the paired end reads, 30 bases, and 10 for the single end reads. If it aligns them in this on this parameter, it will remove those particular bases using the all adapters.fa file. So I have created it. I will show you how I have created it. So just remind me at the end of this session. And then the leading one here, like take the three base quality for the leading ones, and then the trailing quality also take the three here, three spread quality. The main option here for the sliding window for 4 nucleotides at once, take the 4 nucleotides and then check the quality. If it falls below 20, remove that. And then take further 1 nucleotide forward and then take the next three, including the one from the behind, or one from the new and taking the three from the old one, and check the quality. If it falls below 20, remove it. Crop option means I showed you that the quality of the reads was over represented and down represented bases there, percent 80 cg at the ends. So we can select a 245 for the 3' end, and 5' end we will select here 50, right? And minimum length of the reads we can select 50. So if the read length after this trimming and this trimming steps, if it falls below 50, remove those reads. Reads which are longer than 50 base pairs, keep those reads only, right? With these options we can run it. There is the error of output directory. We don't have that directory here. I will create that directory, output directory, this one, Trimomatic out. Now it will run. Okay, it is done. So output is created, those forward and reverse files in the Trimomatic output directory, this one. Here we see forward paired and reverse paired, and then the forward unpaired and reverse unpaired. I did not create those files like log files and then the stat summary file. You can run that on create that file also. You can add those options like when you are running it. you can provide the options like minus log, and then right here run one dot log file, and then minus summary, and then summary run one dot txt. Right, you can create those files to run it. So as we see here, 4 output files. We can run fast QC again on these outputs. So we can run here fast QC minus o mkdir fast QC Tribomatic. So this is the Tribomatic output we are checking now. We can run fast QC minus o fast QC Tribomatic, and then we can provide here all the fast Q files. So forward paired, reverse paired, and then forward unpaired, reverse unpaired. Okay, it is done. Output is created in the fast QC Tribomatic. The same output HTML files, and then the results are zipped also. So I will open these HTML files and then share the screen with you. Okay, here is the forward paired, and then forward unpaired, and this is the forward reverse paired. So this is the forward paired and reverse paired, and then this is unpaired ones. So here is the output from the after the Tribomatic. So we can check here. Quality looks very good right now, which is more than 35, 38 at various positions. So this is the very good quality data now. Then the read lengths. This is the per base sequence quality, mean sequence quality for each read here. So it is also very nice. Right, it goes up here, then drops to 39 for few of the bases here. For few of the reads, not the bases, because it is showing the mean sequence quality score. Then per base sequence content, if you remember that overrepresented bases here, they are now trimmed at this 5 prime and then the 3 prime, and now they are clean. Right, so there is no problem with this per base sequence content here. Then GC content, I told you that time that it is okay when we compare with the theoretical distribution. Then the normally data output, which is more or less, it has these smaller peaks available there or present there, so it's okay for the GC content. Then the per base n content is, it is also okay. sequence length distribution. So it is also showing the same one or same problem here, warning sign that it has the variable length of the sequences, so which is also fine. Rest of the bases which we see here, like the outputs like sequence duplication level, it was okay. Over represented sequences for the forward file, it is now no more present there. So those sequences which were over represented, they were also removed from the forward sequence file. Adapter content was not present there. If we see the reverse file here, which is the which has much better quality. If you see, that time it was going below 20 for the those error bars, but now it is not there, not the case here. It is of very good quality for the reverse reads, and per base sequence content also it is okay. Sequence length distribution, it looks the same as we have for the forward reads. Right, so now the data is much better. I can say it is of good high quality data, and we can go for the sequence assembly, or we can do the variance analysis or any type of the downstream analysis which we are interested in. Right, so this is for the single end files, like for forward unpaired file, and then the reverse unpaired file here. This still has these over represented sequences here, but they are not coming from the any possible source, right, for the single end. And if you see the complexity of this data, which is also very low complexity sequences, like repeats are there, and like CCGG, AG, so low complexity sequences these are. So you can do one thing, like you can remove these sequences or this file even from the downstream analysis for this unpaired. It is not much number of the sequences which are present there, like they have only 927 sequences present in this file, right? So you can omit this file from the analysis. Okay, so this is the how we can check the quality of the data and then process it for the high quality, and then we can again check the quality using the same FastQC toolkit. Right. One thing remains there, how you can create that adapters.fa file. So you can go to the Tribomatic GitHub page. I showed you there at the Tribomatic website. Right. So from there you can go to the GitHub page here. From this page, on this page you will see the adapter folder which is listed here, all the different adapters which are for the Illumina sequencing. So you can select or download all these FASTA files and then put them into single file. So that's how I have created that adapters.fa file. I will send it to you, it will be shared with you, so that wouldn't be a problem. But if you want to create your own, so you can come to the GitHub page of Tribomatic and you can download these sequences from here and add it into a FASTA file, single file, right? So with these few examples, I would like to wrap up this session, and if you have questions, queries, you can ask. I'll try to answer, and then we can close it.